Hello biologists, we're going to do lab 310 here on diffusion, osmosis, and active transport. You should have a lab sheet that looks something like this. As we go through, be looking for these vocabulary words: diffusion, osmosis, passive transport, facilitated transport, and active transport. I'm going to use the software here and we'll get started. So to introduce the idea of diffusion, we're going to ride an air molecule here to understand that all molecules are always moving. Gas molecules in air are moving very rapidly, and diffusion is the idea that all molecules are always moving, and they're moving randomly due to collisions with other molecules. You can see that the definition of diffusion is right here. The natural movement of molecules due to collisions is called diffusion. And hopefully you're getting an idea of how molecules travel and how randomly they travel due to diffusion. The reason we're studying diffusion is to understand how molecules get into and out of cells and how cells come to equilibrium. We're going to start by looking at a really simple model of diffusion. This represents food coloring in the model, food coloring molecules, and how they randomly bump into molecules of water and spread out over time. If you put a drop of food coloring in a real glass of water, you couldn't see the molecules, but you would see the dye, the food coloring, spreading out over time if you didn't stir it. And you might have to leave it there a long time depending on the temperature. But eventually that dye would spread over all of the water and you get a nice uniform color. And that happens because of diffusion. So let's answer the first question here. After looking at the path taken by several red dye molecules, their motion can best be described as moving in a directed wave from high to low, moving randomly through collisions in no particular direction, staying close to other red dye molecules, or moving in clumps of molecules together. I'll let you decide what the best answer is. It's often said that substances diffuse from highly concentrated areas to area of low concentration. Describe how this works if a molecule can't tell that it is in a region of high concentration and just diffuses randomly through collisions. So let's stop this model here, and you can see how diffuse or dispersed the dye molecules have become. Let's reset it and do it over again and just place a drop. Let's put it in the middle this time. And you can see if you watch just one dye molecule, it doesn't randomly, it doesn't head for the sides of the container. It's just bumping into everything. And it goes back and forth and back and forth and maybe to the middle and maybe to the edge and then back again. But eventually, since there are a number of molecules here and everything is happening randomly, they're going to spread out. Just because it's random that they would not all stay together. It's more random. Nature always becomes more random if given the chance. Okay, biologists, so at this point, you should be able to answer question 1 and question 2 on your worksheet. Now we'll progress down the worksheet and talk more about diffusion and how it affects cells. So the next big idea or big question we're going to talk about is, how does concentration affect the overall flow of ions and molecules into and out of a cell? All cells need oxygen because they do a process called respiration. They need that oxygen to do chemical reactions with simple sugars like glucose. And a waste product of these reactions is carbon dioxide. So it's very important if we understand how oxygen and carbon dioxide get in and out of cells, because that's essential for us being alive. Below is a model that we're going to look at, divided by a cell membrane. On the left hand is the liquid outside the cell. You can see in the blue here. And on the right is the liquid inside of the cell. Oxygen and carbon dioxide levels are shown by these little bars once we start to run the model. We're going to experiment with how concentration will affect the overall movement of substances in and out of the cell. Let's start with kind of a real situation. There's a lot of carbon dioxide in your cells because it's a byproduct of the reaction to break down sugar and make ATP. And there's a low concentration outside your cells. Let's run the model and see what happens over time. Look at the concentration outside and inside and watch the molecules. Watch how many are heading inside to outside and watch how many are heading outside to inside the cell. After a while, you can see that the concentrations are becoming much more equal. And the cell reaches a state of what we call equilibrium. It doesn't mean that everything has stopped moving. It just means that the direction from inside to outside is equal to the rate of outside to inside. So they're moving back and forth at the same rate. Let's set up another situation. I'll get rid of the CO2 here so it's easier to see. And we'll just look at oxygen to make things simpler. So the O2 concentration outside your cell is usually pretty high because your red blood cells are going to your lungs, picking up oxygen from you breathing, and we'll talk a little bit more about that later, and then they're kind of dropping it off, that oxygen, at your cells, which need it to do photosynthesis, to do cellular respiration. So let's run this model with high oxygen on the outside of the cell and low oxygen on the inside, and focus on which direction the molecules are coming most often. Some of them are going inside to outside, but probably you'll notice there's more coming from outside to inside until you get everything even. So let's look at this next question. Set up the model so that there's a high concentration of something outside and a low concentration of that same thing inside. Well, I'll reset the model because that's just what we did. What are the chances that a molecule will move into or out of the cell? Is there a greater chance that the molecule will move from lower concentrated to higher? That would mean that things are just going to get higher and higher on the high concentration side. Is there a greater chance that a molecule will move from higher concentration to lower concentration? That means things will even out. Or is it the equal chance that a molecule will move in either direction? I'll let you think about that one. The next question says, describe the flow of molecules when there is an area of high and low concentration. And I'll let you read the options. They're on your lab sheet. So at this point, you should have been able to answer question 4 and question 5 on your lab sheet. So in this section, we're going to talk about what is equilibrium and how does it relate to diffusion. So we're going to be talking more about this idea of the concentration on both sides of the cell membrane becoming equal. That's what equilibrium is. But it's a dynamic equilibrium. It doesn't mean things just stop. It means the rate going one way is equal to the rate going the other way. Nature always tends towards equilibrium, but it always keeps moving. So let's see some different combinations and see if you can tell when the model reaches equilibrium. So let's look at a kind of true to life scenario. We're going to have high carbon dioxide on the outside. We're going to have low carbon dioxide on the outside. We're going to have high oxygen on the outside. And on the inside, we're going to have high carbon dioxide because that's a waste product your cell needs to get rid of. It's a waste product of producing ATP. And oxygen is something your cell uses up making ATP, so it's low in oxygen, needing some more oxygen from outside the cell. So we'll let this model run and we'll see what happens. Watch the purple And then watch the yellow. Remember, the yellow is oxygen. And the purple is CO2. And look at what's happening across the membrane. Things are moving and moving and continue to move, and they're moving in both directions, and everything's bumping into each other. The carbon dioxide and oxygen are bumping into each other. And remember, diffusion is the random movement of things based on collisions, that it becomes random because of collisions. And eventually, if we wait long enough, we can see that the concentration of carbon dioxide is starting to level out on both sides, and the concentration of oxygen is starting to level out on both sides. So let's go down and see if we can answer the questions. What is true of the concentrations when equilibrium has been reached? Well, we're getting close to equilibrium, so you can kind of think about that. What is true of the rate at which molecules move into and out of the cell at equilibrium? I'll just give you this one. At equilibrium, it's really, really important to understand that things don't stop moving, that they move into and out of the cell at the same rate. In other words, they're going this way at the same rate we're going this way. So at this point, you should be able to answer question number 6 and question number 7 on your lab sheet. So in this section 4, we're going to ask ourselves a really important question to our health. Why do red blood cells, which are in your blood, absorb oxygen in our lungs and release it to the rest of the body? Why doesn't it happen the other way around? Why aren't our cells, our red blood cells, absorbing oxygen from the rest of our body? Let's go down and look at a red blood cell to find out more. So here's a red blood cell in an oxygen environment. It's right around your lungs, and it's in equilibrium. The concentration of oxygen on the inside is the same as the concentration on the outside. Now, if we take this red blood cell on a journey down through your blood vessels, down to your toes, where there's not very much oxygen, because your cells have used it up, what direction is the oxygen going to head? Are things going to come to equilibrium? Let's do that one more time. Let's go back up to the lungs, pick up some oxygen. So we're going to an environment where it's high oxygen on the outside and low on the inside. It quickly comes to equilibrium, and then the cell takes a little ride through your blood vessels down to your toes where the oxygen's pretty low, because you've been making ATP, and the oxygen on the outside is leveling off with the oxygen on the inside. Oxygen is leaving the red blood cell and going to your toes. If we take it back up, oxygen is going to head into the red blood cell from your lungs. So this is a high oxygen environment near your lungs. This has been a low oxygen environment near your toes or like your working muscles. Anything that's using up oxygen pretty quickly is a low oxygen environment. So let's answer these questions. What happens to the oxygen concentration of the cell when you move it into a new environment? Should be able to answer this one from studying the model. Explain how a red blood cell delivers oxygen from your lungs to the rest of your body. Well, we can look at the model one more time before you answer this question. So if we take it to your lungs, it's in a high oxygen environment. There's low oxygen on the inside of the cell, but quickly it comes to equilibrium. The red blood cell fills up with oxygen. Then when we take it on a ride to part of the body where there's a low oxygen concentration, because the cells are doing cellular respiration and making ATP, that oxygen diffuses out of the cell and into your tissues. So out of the red blood cell and into your, say, your muscle cells. So at this point, you should be answering number 8 and number 9 on your lab sheet. So in this section, we're going to talk about hemoglobin. Red blood cells have evolved to absorb more oxygen that can normally happen through diffusion, they get a little help absorbing extra oxygen by using this protein called hemoglobin. Let's find out more about hemoglobin. When a red blood cell enters your lungs, it enters the most oxygen-rich part of your body. So the concentration of oxygen outside the red blood cell is highest. We learned that in the last section. However, the amount of oxygen you can get into a blood cell using the natural concentration difference inside and outside the cell in the lungs is not enough to supply your body with all the oxygen it needs. For instance, right now your brain is using a lot of oxygen because you're thinking. Brains are oxygen-hungry organs. So animals, fortunately, have evolved an answer to get more oxygen into their bloodstream. Red blood cells are special. They're packed with hemoglobin. Here's the hemoglobin molecule. It is made out of 4 subsections of amino acids that are strung together, take shape, and then they come together, these 4 parts, to make a hemoglobin molecule. And they all carry a heme group, which has iron in it, and it hangs on to the oxygen more tightly than the cell would with just diffusion. So let's go down and see what happens when we add hemoglobin. So let's look at it without hemoglobin. We'll just run it. The cell is going to absorb some oxygen, and it's just going to come to equilibrium with its environment. Now, if we give it some hemoglobin on the inside of the cell, these stand for the hemoglobin, and we'll run it. Each of these hemoglobins can absorb 4 different oxygens and hang on to those oxygens. So you can see the different heme groups, 1, 2, 3, 4. That means each hemoglobin is going to hang on to 4 oxygens, plus oxygen is just going to diffuse into the cell. So you can see that the concentration on the inside of the cell is a lot greater than the concentration on the outside of the cell. So it gets a boost. It's more than just equilibrium with hemoglobin. Hemoglobin helps it boost the concentration of oxygen in the cell higher than it would be just with equilibrium. So describe what happens when the red blood cell contains hemoglobin. Well, hopefully you know by now. And you should be able to explain how hemoglobin helps transport more oxygen that can normally be done with simple diffusion. There's a clue here. Look at this level here of oxygen concentration. And look at hemoglobin kind of hanging on to a whole bunch of oxygen so that the concentration of oxygen inside the cell is greater than outside the cell. In other words, you get to kind of cheat equilibrium by using hemoglobin. So at this point, you should be able to answer question number 10 and question number 11 on your lab sheet. In this section, we're going to talk about what role does surface area play in diffusion rates and why is the diffusion rate important? A diffusion rate is how fast oxygen can get across the cell membrane. And if we want to breathe, if our muscles are using a lot of oxygen to make ATP and help us move, we need to get oxygen across the cell membrane into our muscles or into our brain if we're thinking really hard, so that how fast we can get oxygen is the diffusion rate. So one theme you'll see in many biological systems is a network of channels that form smaller and smaller branches. You can see that in your lungs. You have a trachea here and it gets a little bit smaller and then gets smaller and smaller and smaller and tinier until it gets to the alveoli, which are tiny little kind of sacs that line up with some tiny blood vessels where your red blood cells can interact with the oxygen coming into your lungs. Our lungs, our blood vessels, our kidneys, to name a few, are these branching systems. There seems to be an evolutionary advantage to having these branch systems, which greatly increase the surface area between the inside and the outside of these structures. If you spread out the surface area of all the little alveoli, these little sacs in your lungs, you would have an area the size of a tennis court. So imagine taking a tiny, like a sheet of something that's the size of a tennis court and trying to stuff it into your thoracic cavity, the part of your body from here to here where your lungs are. That'd be a pretty difficult job, but your lungs do great work. To the right, you can see how the air passages in your lungs branch out into smaller and smaller branches, ending in alveoli. Why have we evolved to have such delicate, high surface area systems? What advantage is there to having a large surface area separating two things, like the inside of the lungs and the bloodstream, the inside of the digestive system and bloodstream, and the inside of the kidney and bloodstream? Your digestive system, the area, because it's got all these little fingers in it, is the size of the square footage of a McMansion, about 3,000 square feet. So there's a lot of surface area in your digestive system too. Let's use the model below to figure out how surface area affects diffusion rates of a substance across a boundary. So this is a small surface area. You can see we can adjust the surface area here. Let's run it with a small surface area and we can kind of see how long it takes to get to equilibrium. We're waiting for these two to be even. And I like to make an analogy here. This is kind of like being in the Xcel Energy Center or the US Bank Stadium in a really big arena and only having one door. It's going to take a long time for people to get in and out of the building. Let's get some more doors or some more surface area, some more room for things to go back and forth and in and out, and run it this way. And then let's compare that to the time it took before. It seems like it's going a little bit faster this time, just for things to even out. You know, this is maybe having like 5 or 6 doors in a big arena instead of just one. Imagining 10,000 people trying to get out one door. It would take a really long time. If you give them a couple more doors, a couple more places to get in and out, things are going to run faster. And if you give a cell a really large surface area, this is kind of what the inside of your intestines look like, there's a lot of places for things to get in and out. There's a lot of doors, and this is kind of more the reality of a large arena. They've got a lot of doors, so if people need to get in or out quickly because of some emergency, they can. You can see it comes to equilibrium very quickly. We go back down to a smaller surface area, it takes a longer time. So the more surface area you have, the more quickly things can diffuse. There's more places for things to get across, kind of like having more doors. So how does surface area affect diffusion rates? I'll let you answer this one. So single-celled organisms absorb everything they need directly through their skin, their cell membrane. However, you could never get enough oxygen if oxygen could only diffuse through your skin. Explain why it is necessary to have lungs with large surface areas. Well, we know that the more surface area you have, the faster things can diffuse because there's more places things can go across the cell membrane. If there's more cell membrane, there's more places to cross over, just like having more doors on a stadium. I'll give you a hint, this is not the answer, but our skin on the high end of things runs about 21 square feet. That is, just an estimate for the population at large. That's your skin. Now, the surface area of your lungs on the high end of the range for the general population runs 110 square feet. So there's a big difference. We have a lot more surface area in our lungs, and why do we need all that surface area? You should explain using lungs, the words lungs and surface area and diffusion rate in your answer. So at this point, you should be able to answer question 13 on your lab sheet. There are some hints in these screenshots. And you should also be able to answer question 14.